All right, we'll get started for today. A couple of things for everybody. Make sure you register for um, your presentations. Okay, so you're gonna go all the way down. You're gonna look for schedule for presentations, and then you're gonna come and click on this link here at the very top. And again, this will take you to the presentation schedule, right? There's very few of you who have actually registered, and we start presenting on November 23rd. Okay, so that's Monday, right? Um, so please be sure to register, okay? And again, this is going to be the presentation that is on your paper, or the topic will be about your paper. It's just going to be a 10-minute spiel about everything you've learned, the research that you've done. You could have a PowerPoint, you can have an aid, but please just make sure you register for the date, because there's not very many time slots, okay? And this is points you're gonna miss if you don't have a little presentation, okay? So if you don't have anything scheduled, you don't present, right? So be sure to sign up for a time slot. Again, it'll be during lab. I will have a Zoom link that I'll send out to those individuals who uh, presented that day, or I'll actually send it to the whole class. But it'll be for those people on that day who signed up will come in during those times, okay? Um, and you present. So please just make sure there's very few people that actually have signed up. There's 12 people that have signed up, this is none, okay? So please, if you're having issues with that, let me know. We can try to help you. But if you don't know what's going on and you're in the class, you please just make sure that you schedule your presentation, okay? I'll be sending an email out to kind of remind people, but just be sure that you're scheduling that, okay? All right, we're going to finish with chapter 26 and get into chapter 27. All right, pregnancy and early development. Pregnancy is the carrying of one or more developing embryos in the female uterus, also called gestation. Stem cells are relatively unspecialized cells that can give rise to one or more types of specialized cells, right? So this is where that huge debate comes with government and research, right? Stem cell research. There's a lot of issues that people have with working with stem cells, right? They don't want anyone interfering with life, and especially with fetal stem cells, right? There's two different types of stem cells essentially. There's pluripotent and totipotent stem cells, and essentially they don't want to be messing with the stem cells that are harvested from fetuses, okay, or from the mother's placenta, okay, or after birth, right? They want most people think stem cell research occurs at that level, whereas everyone has stem cells. We have stem cells in our liver. Um, there's been research done with using the stem cells in adult humans to regenerate a nervous tissue, right? So there are other different types of stem cells. Okay, there's not just stem cells that you find in fetuses or in afterbirth. Okay, placenta. In most mammals, the organ that produces nutrients and oxygen to the embryo and helps dispose of metabolic waste. The placenta is formed from embryonic tissue and the mother's endometrial blood vessels. And we'll see what a placenta looks like now, okay, or in a little bit. So early stages of development, most of the early stage development occurs in the oviduct, okay? So fertilization occurs in the oviduct, okay? So after the follicle releases the egg, okay? Um, so the secondary oocytes traveling, it gets met with sperm. Becomes a zygote at this point, okay? And then it has multiple divisions of cells, and then the blastocyst is what actually implants in the endometrium or in the uterus, okay? So this is what actually implants in the rich blood vessels of the uterus, and this is the start of the placenta. This is a start of the attachment to the mother, okay? So here we have inner cell mass, And in here, the red occurs at day 6, the implantation. Okay. At day 10, we have multiple cells maturing and multiplying, and this is going to be turning into the placenta. Okay, this is the attachment site of where the child's going to be exchanging nutrients and waste products. This is what's going to be attached through the umbilical cord to the child eventually. Okay, the actual size of the cell mass is actually shown here. Okay. So at day 10, after implantation, the inner cell mass, the embryonic stem cells, will develop into a fetus. The outer layer of trophoblast cells will become the embryo's contribution to the placenta. Okay. So here you have the blood vessel innervation. So you have the outgrowth of veins and arteries to the placenta, and you have multiple areas of capillary beds. Again, this is going to be exchanging um, nutrients as well as waste products from the fetus or the embryo. Okay. Chorionic villi. Again, this is going to be the area where you have, think of these as capillary beds. They're going to be exchanging gases and waste products to the intersagittal fluid, okay, of the child. But eventually, this will lead to the umbilical cord or to the fetus of the child. Okay. So here we have after 35 days, this is what the fetus will look like. So 5 weeks, 9 weeks, we start having the development of what look to be hands and human features. At 14 weeks, it looks almost like a complete with separated fingers. Uh, Eyes are forming. And then at 20 weeks, you see nearly a full child, like nearly a full premature child here. Again, very um, early on, and then birth is 280 days. Again, fully formed child. But you can have in the third trimester, the start of the third trimester, about a week or two into that, the child can technically be born. Still needs a lot of help, but again, it's a fully formed fingers and toes at that point. At the second trimester, actually, you'll start having the formation of the full fingers and toenails and things like that. So full development at that point. Ultrasound. So an ultrasound image is produced when high frequency sounds are passed over the abdominal cavity. Look for the child. And again, this is to check the health of the child. It's good to use to check for the heart condition, any other type of conditions such as spina bifida or abnormalities can be seen within the fetus as well. Okay. Childbirth or labor is a series of strong rhythmic uterine contractions that expels a baby out of the uterus and vagina during childbirth. So remember, the cervix does need to dilate, right? There needs to be an opening of for the child to pass through. And again, those contractions, those muscle contractions, again, are for the opening of the vagina and the cervix for the child to pass through. Okay, and these are incredibly taxing on the mother. Okay, I'm not a female. I wouldn't know, but from what my experiences, from what I've heard from females, is that the contractions actually hurt more than the actual childbirth itself. That the contractions are so intense that um, that's what the pain is attributed rather than the actual pushing out of the child, but the contractions during the expansion to open up for that child to pass through the birth canal. Okay, so during birth, the child needs to be facing head first, right? In this manner. A breech birth is actually the inversion of the child, so the foot first, which is difficult, right? The largest part of the child will be the head. The bones in the child's skull are not fused yet, and many of you probably been around babies. They do have a soft spot, right? And that's because the skull is not fused entirely, all the sutures. Um, and this is to allow the child to pass through the birthing canal, right? You do have bone here that can limit the amount of space that child has to pass through. Okay, um, and if a baby is too large, it may or may not be too difficult for that child to pass through the pelvic girdle. Okay, And women do have a larger opening than men for their pelvic girdle, right? And that occurs early on in development for the female, and it's just an angle of how you have the pubic bones fused, okay? And this allows for the child to pass through, and this is why people that go back and they date fossils, they look at fossils, whether it's a male or female. You can do this genetically, but you can also do this just by looking at the pelvic girdle. Okay, if you look at the pelvic girdle, you can see the tilt and the opening um, of that birthing canal, and you can tell right away that it's a female. Okay. Infertility, so the inability to conceive after 1 year of regular unprotected intercourse. So this is actually defined. I didn't even know it was defined. I thought you would need a diagnosis of a sperm count or non-viable egg to actually um, be infertile. But infertility is sexual intercourse between two people for a year. Apparently, it's the definition. Impotence. This is the inability to maintain an erection, also called erectile dysfunction. So typically, impotency in males is associated with lack of blood flow. So an unhealthy elder male, or elderly male, will typically have impotency. It can also be caused by other factors such as nerve dysfunction or even stress. Okay, um, but typically, what you see is elder males that have atherosclerotic arteries, or they are very unhealthy, high blood pressure, um, things like that. Okay. So a switch from briefs to boxers improves some cases in fertility. Um, and this is mainly associated with cooling of the testicles. Okay, so testicles or sperm needs to be cooled down to a little bit below what body temperature is. And this is why the scrotum is there, right? The scrotum will relax and allow for the testicles to sit outside of the human body. You'll Men will notice in the winter that the testicles will actually become closer to their body, or move closer to their body if it's too cold, right? If it's too hot, the scrotum actually relaxes and allows the testicles to hang further away from the body in order to cool. Um, but a lot of instances of fertility can be attributed to the type of underwear you wear, right? Especially fertility doctors, will do the best they can to try to limit the amount of sperm being destroyed either by heating or by activity, right? They talk about also cycling being very difficult on the testicles as well. So the physician may recommend that the individual does not cycle while trying to conceive. Okay. And I'm talking about cycling for hours, like Lance Armstrong. Those individuals who are riding up to the mountains on their bikes. Okay, really a lot of stress on that area. Okay. All right. So in vitro fertilization. So this is going to be the artificial insemination and fertilization of an egg. Okay. So you can harvest an egg from a female, harvest sperm from a male, and um, essentially fertilize the egg in a petri dish. Okay. After this development, you have an early embryo. You can implant this embryo inside of a female in the uterus. Okay. You can also have a surrogate mother. There have been instances where people will take up surrogacy, so they'll have an egg from the female who cannot conceive, and the male, or the father, and then they have a third party or woman come in and actually have the child for them. Okay, but this can be performed this way as well. And again, that baby, that child, is genetically identical to the parents, not the woman giving birth. Okay, the woman giving birth is suffering through all the birthing pains or issues, but that child is genetically going to be related to the donor of the egg and the donor of the sperm. Okay. All right. That's it for chapter 26. All right. We'll get through about half of 27, I think, because it's a little large. It's a little larger of a chapter if we get through it. All right. So nervous, sensory, and locomotor systems. So an overview of animal nervous systems. Nervous system, the organ system that forms a communication and coordination network throughout an animal's body. A neuron, a nerve cell, the fundamental structure and functional unit of a nervous system, specialized for carrying signals from one location of the body to another. Okay, so nervous systems can be the central nervous system, the peripheral nervous system. So central includes the brain and the spinal cord. The peripheral nervous system will include the various ganglion and the other neurons that are descending away from the central nervous system. Okay, it can also be referred to the parasympathetic nervous system. Okay, which we'll get into in a little bit. A neuron. So one important trait of neurons is they don't regenerate. Okay, so this is why it's important not to damage, not to destroy brain tissue or neurons. It becomes very difficult to repair. They have done research with stem cells from the liver or elsewhere and trying to replace neural connections. And they've had some success with this, but neurons essentially do not divide anymore. That they're there for your life. If you damage or destroy or kill the neuron, then you lose the neurons. Okay, doesn't mean that you're not able to have neuroplasticity, or it doesn't mean your brain's not able to make new connections. It just means that you're limited on the number of neurons you have. Okay, so take care of them. Okay, so dendrite. So this is a short branch neuronal fiber that receives signals and conveys them from its tip inward toward the rest of the neuron. We'll see this right now. We'll see the directionality of both dendrites or axons. Okay, so an axon is a neural fiber that extends from the cell body and conducts signals to another neuron or a muscle or a gland cell. Okay. Synaptic terminal is a bulb-like structure at the tip of the transmitting neuron's axon, where signals are sent to another neuron or to a muscle or gland cell. Myelin sheathing, think of this as almost a rubber coating on your electrical cord that you have in your house, right? Let's say you're charging your phone. You have that electrical covering over the wire. This is what myelin sheathing is used for. It's for that insulation to allow for that electrical signal to travel down that wire. Okay, if you don't have that sheeting, you don't have that cover, you lose the electrical charge, right? So a chain of bead-like supporting cells that insulate the axon of a nerve cell in vertebrates. The insulation helps to speed electrical transmission along the axon. Okay, this is where you have MS or multiple sclerosis. Okay, this is the destruction of that by your immune system, and we'll talk about that right now. All right. So the directionality is the dendrites receive signals through the axon or the terminals. So there's other axon terminals here from other neurons, and it essentially sends the signal to the dendrites. The dendrites will pass this to the nucleus and the control center of the cell. This cell will determine whether or not to send the signal. Okay, there could be some stimuli that is not sent down this terminal. Okay, so here we have the axon and the synaptic terminals. So other synaptic terminals are binding here to these dendrites that receive the signal, right? In order to send this signal, you start an action potential here. And what this means is almost like an electrical charge. It's literally a shock wave that passes through this axon terminal. This myelin sheathing, these are actually cells. These cells insulate that electronic signal or that signal that's being passed down through the axon. Okay, without the myelin sheathing, you lose the signal that's being passed to the synaptic terminals. Okay, in MS, individuals actually mount an immune response to these cells attached to the axon. What that means is that your immune system is actively attacking the myelin sheathing or these cells that make up the myelin sheathing. Therefore, you lose the conductivity in the axon fiber. Okay, this means you don't send the electrical signal. This means you have issues, nerve issues associated with those individuals. Okay. All right. So central nervous system, so CNS, the integration and command center of the nervous system, the brain, and in invertebrates, the spinal cord. Okay, so brain and spinal cord make up the CNS, right? Peripheral nervous system is a network of nerves carrying signals into and out of the central nervous system, right? So these are all those nerves that are coming out of the foramen in the spinal cord. They are located outside of the central nervous system, okay? So sensory input is the transmission of signals from sensory receptors to integration centers in the central nervous system. So sensory neuron is a nerve cell that receives information from sensory receptors and conveys signals to the central nervous system, okay? So sensory input and sensory neuron, right? Integration is the interception of sensory signals and the formation of responses within the nervous system, okay? Interneuron is a nerve cell entirely within the central nervous system that integrates sensory signals and may relay command signals to motor neurons, okay? And we're gonna take a look at these in a minute. Motor output is a process of sending signals from the central nervous system to the peripheral nervous system that results in a response such as movement. So again, this is going to be CSN outward. So this is me telling my hand to move or pick up or type, right? Motor neuron is a nerve cell that conveys command signals to the central nervous system, from the central nervous system, rather, sorry, to receiving cells such as muscle cells or gland cells. Okay, motor output is an action. Motor neuron is a neuron that's going to be performing that action for those muscle cells. Okay. And here's a nice little diagram kind of depicting that, the organization of the nervous system, right? You have a stimulus, we have sensory input, we have integration, and then we have output, right? So think of the stimulus as a mosquito on your skin. I feel the mosquito, sensory input integrates that, right? And then all those dendrites will decide what to do with that signal. The dendrites will send out that signal through the axon into the synaptic terminals. That'll then be an output, which is a smacking of that mosquito, hopefully, as the output occurs. Okay. So stimulus on skin. This is a peripheral nervous system going into the central nervous system. We have that processing. Then we have that motor neuron output. Okay, which is going to be the motor neuron telling your hand to smash the mosquito. Okay, this is a very delicate and intricate process that occurs very rapidly. Okay, it's very important that your body is able to pick up on stimuli and utilize the stimuli in order to have an output or a response to that. Okay. So action potential. So resting potential is a voltage across the plasma membrane of resulting neuron, positively charged outside, excuse me, and negatively charged inside. Okay, so action potential, this is a self-propagating change in the voltage across the plasma membrane of a neuron, a nerve signal. Threshold is a minimum change in a membrane's voltage that must occur to generate a nerve signal. This is the action potential. This is what I was referring to earlier. Okay, so generation of an action potential, right? So we have a resting potential. So this is going to be the negative charge on the inside, and on the outside you have that positive charge. Okay, so this is non-firing. This is ready to go. Okay, stimulus opens the first set of ion channels of if the threshold is reached and action potential is triggered. So depending on whether or not that nerve has been stimulated enough or well enough will determine whether or not the action potential of the nerve is going to fire. So in all of your nerves, you have these positive and negatively charged ions that are separated by the membrane. Okay, so think of this as almost like your mitochondria, right? There's separation. This is an electrical chemical gradient, right? We talked about that before with the proton. This is mainly salts, though. So this is just an electrical gradient, right? So just charged particles that are separated. If a stimulus occurs and it triggers the action potential means it's exchanging of positive and negative charges, then you have a nerve impulse, a nerve signal. So here you have the positive ions rushing in, and this is going to be triggering the action potential, the nerve impulse. Ooh, excuse me. Additional channels open in the region of the neuron, and the interior of the cell becomes more positive than outside. Okay, so what happens is you have an electrical change. You have the rushing of the positive to the inside of the cell, those positive charge. This creates an electrical current. Okay, so the set of ion channels closes and inactivates. A second set of channels opens and positive ions rush out. The membrane returns to resting potential. Okay. Um, so this is going to be displaying the resetting of the nerve, right? So your nerves need to reset after they fire. But again, this propagation of the electrical current is done by the flipping of the charges, right? This is an electrical gradient. Okay. positive and negative charges. Okay, so think of this as a domino effect. Okay, so essentially what happens is when the action potential starts, you have this a rushing in of positive charge. This flips the charge for a second. That immediately changes back, and the next positive charges, or the next gates open up, and then they revert back. Then another propagation occurs, opening up, and then goes back. But this again is triggered by the electrical impulse, the electrical signal. Okay, so boom, they rush in. You have the positive charge. They flip back. This encourages this to start the action potential and finish it. So boom, and this happens rapidly. Okay, um, so you have the action potential starting. So this is the new action potential being started. This is going to be reverting back to resting potential, and then you have essentially the propagation. So I take like the dominoes is a good explanation, but it's almost like I think of the flipping of the light switches, right? So it's on, off, on, off, on, off, on, off. And this is essentially going down the row and turning on or off the electrical impulse. So this is on right here. This will eventually go back to off. So this can turn on, and it essentially just passes down the signal through that axon. And it's very fast. It's rapid. Okay. Um, but again, think of the rushing in of the positive charge is starting that electrical signal. This triggers this to switch the next action potential, and then boom, it just keeps passing through, keeps going down that line of that axon. Okay. So passing of signal from a neuron to a receiving cell. We have a synapse. This is a junction or relay or a relay point between two neurons or between a neuron and a muscle or gland cell. Electrical and chemical signals are relayed from one cell to another at a synapse. We have a synaptic cleft, which is a narrow gap at a synapse separating the synaptic terminal of a transmitting neuron from a receiving cell. I know it's a lot, apologize. Which is another neuron, a muscle cell, or a gland. Okay, so we also have a neurotransmitter, which is essentially acetylcholine, a chemical messenger that carries information from a transmitting neuron to a receiving cell, which is another neuron, muscle cell, or gland cell. Okay, and we'll see that right now. And again, this is occurring at the synaptic terminal. So this is done after the action potential has been completed, right? So after all of these electrical currents are sent down the axon. And remember, we have the myelin sheathing here on the outside that helps insulate and helps send this current down. Upload a video that I feel like there's a video that better explains and shows how these motor neurons fire. Okay, so I'll upload a YouTube video so you can see that. Okay, but again, the myelin sheathing are at either end. Each of these gates is actually in between a a Schwann cell, which makes that myelin sheathing. Okay, yeah. All right. So at the tips, at the axon terminals, you do have the dumping of the neurotransmitter that's being released. Okay. So typically we think of acetylcholine, right? This is being dumped into this open space, this terminal that is touching another neuronal cell at the dendrites, right? So this dendritic region, you have these open ion channels that'll bind to the neurotransmitter and allow for the transmission of an electrical signal or that new signal, okay? And it's up to this dendrite whether or not to send the signal away, okay? If one signal is not enough to activate that specific dendrite, then multiple need to come and actually activate it. Multiple signals need to be reached by this dendrite in order to have that action potential reached, right? And it's really up to that dendritic cell of the receiving end to send that signal, okay? And there's different thresholds, right? What that means is there needs to be a certain criteria that's met for the amount of signals, the amount of neurotransmitter that's being dumped onto this dendrite cell or dendritic cell, or this there's neuron in the dendritic region. Okay, so there needs to be enough neurotransmitter. There needs to be a strong enough, a strong enough sign or signal from other cells in order to transmit that electrical signal again. Okay. So here we have multiple synaptic inputs, right? So in order for that threshold to be met, there needs to be multiple instances of activation. Okay, and this is what's going to eventually lead for the action potential to be reached, right? And so you see the myelin sheathing here. This is that cell, that Schwann cell. It's going to be making up the myelin sheathing. But for this action potential to be reached, multiple signals need to be met. Then they can start firing off that electrical impulse. That electrical impulse essentially starts here. And then you have another electrical exchange here, right? The positive goes in, the negative goes out, and then it gets flipped, right? And again, this electrical signal can be sent quickly because of the myelin sheathing. This can be transferred here, boom, this gets transferred there, boom, and so on and so forth. And these are just an image of the synaptic terminals. Yeah, let me see, because I think it's a better way to explain. Yeah. So I'll tell you this. This action potentials that you see here, the activation of the action potentials, each of these gates, this positive and this negative shifting, occurs at breaks in the myelin sheathing. So a positive and negative flip will happen here, positive and negative flip will happen here, positive and negative flip will happen here. So this essentially limits the amount of time needed for the electrical signal to travel, right? The electrical signal travels here, boom, activates, travels here, boom, activates, travels here, boom, activates, travels here, boom, activates. So at these individual spaces is where you have the electrical impulse being sent to the synaptic terminals. And again, in order for another dendritic or a nerve cell to transmit that signal, multiple axons need to be activating that dendrite. It needs to meet a threshold, needs to meet a certain amount of activation, a certain amount of neurotransmitter needs to be provided to send that signal away again. Okay, because some stimuli does not warrant a response. Okay, not everything requires our central nervous system to process and react to it. If it did, then we would be very, very exhausted and running out of neurotransmitter, right? So not everything you react to, and that makes sense because that's going to be just too much. And this is just showing an or synaptic terminals binding to a dendrite, the dendritic region of a neuron. Okay. All right. Let me see what time it is. So the central nervous system, the CNS, of humans includes the brain and spinal cord. The peripheral nervous system, which is PNS, consists of nerves that convey information from the CSN. Okay, so here we have the peripheral nervous system, right, which is everything else that is not included in the spinal cord and the brain. Okay, so these neurons that are extending from the back, from the foramen, again, are the PSN, the peripheral nervous system, whereas the central nervous system is the spinal cord and the brain. Okay, so the central nervous system, which is essentially integration. We're talking about a couple of the organs and the structures. So we have the brain. This is a master control center of the central nervous system, which is involved in regulating and controlling body activity and interpreting information from the senses. Okay, spinal cord. In vertebrates, a jelly-like bundle of nerve fibers located within the vertebral column. The spinal cord and the brain together make up the central nervous system. Cerebral spinal fluid that surrounds, cushions, and nourishes the brain and spinal cord and protects them from infection. Okay, so very important. Then you have the meninges, layers of connective tissue that enwrap and protect the brain and spinal cord. Okay, so here we have a diagram of the brain. We have the cerebral spinal fluid shown here being secreted in the central brain cavity. So this is leaked out and gets produced inside of the brain, and these horns here, and essentially the horns will leak or secrete the CSF that covers the brain and the spinal cord, right? And again, the production of the cerebral spinal fluid occurs in the brain. Okay. And you have the spinal cord as well encased in the brain. So if you've ever heard of a spinal tap, or an individual needs to see if to check their spinal fluid, they get a spinal tap actually in their spine. So in their upper spine, not really close to their brain, but lower spine, and they go and they inject, and they suck out some CSF or cerebral spinal fluid. And this is what they're collecting. And this is a clear fluid. It should be clear. You shouldn't have any pigmentation or color to it. Very few cells are present in that CSF. Um, Many times the cells you do see when we do an analysis on the CSF is actually red blood cells that leak in from other areas. Okay. This is a very sensitive task, right? You don't want to be damaging any nerves. Motor system. This is a component of the peripheral nervous system. Vertebrates composed of neurons that carry signals to skeletal muscles, mainly in response to external stimuli. The motor system is primarily under voluntary control. Okay. Autonomic nervous system, a component of the peripheral nervous system of vertebrates that regulates the internal environment. Okay. The autonomic nervous system is made up of the sympathetic nervous sympathetic and parasympathetic subdivisions. The autonomic nervous system is primarily under involuntary control, right? Fight or flight. Okay, this is going to be those the nerve signals that control really the fight or flight, right? Consuming of food or breaking down of food, or fighting. Okay, or getting ready to have a reaction, right? We talked about us adrenaline being used to stimulate this response, the fight or flight response, in order to prevent an allergic reaction, to shut down the immune system, right? So parasympathetic division is one of two sets of neurons in the autonomic nervous system, generally plays long-term functions, such as obtaining and conserving energy. So think of rest and digest, okay? This is going to be the immune system, peristalsis in the intestine, right? So breaking down of food. So parasympathetic division is rest and digest. So protect your body with the immune system, as well as perform digestive functions in the smooth muscle in your GI tract. Sympathetic nervous system is the opposite. So one of the two sets of neurons, blah blah blah. It generally prepares the body for energy consuming activities, fight or flight. Okay, and this is going to be important, right, when we activate the sympathetic nervous system with adrenaline, right? This will stop the parasympathetic nervous system, right? So adrenaline kicks this off. It stops the immune system, right? This is why we have EpiPens. EpiPens shut the parasympathetic division off and activates the sympathetic in order to stop the immune response of the allergic reaction, to stop the closing of the upper respiratory tract, to stop the allergic reaction, or the hives, or any type of rash you might get as well. Okay, so it literally shuts down the immune system. All right, so here's some major parts of the human brain. We have the cerebrum, performs sophisticated integration. So these are higher level understanding, especially with the cortex. So we have the succuli and the gyri here. So the individual little accumulations in the brain. The brain is shaped like this because it allows surface area, right? So the reason why the brain is not smooth is because we need surface area for the neurons. Okay, so think about your microvilli in your intestine, right? You have more surface area in your gut to essentially collect more food. The brain has more surface area in order to have more neurons. Okay, in order to have more neuronal connections, that allows for more surface area, right? More area to have neuronal connections. All right. So outer layer of the cerebrum involved in memory, learning, speech, emotions, formulates complex behavioral responses. Okay. You have the hypothalamus. We talked about the hypothalamus being integrated with the pituitary gland. So regulates autonomic nervous system, serves as a homeostatic control center, controls pituitary gland as a biological clock. Okay. Um, we have the brain stem, which is made of the midbrain, pons, and medulla oblongata. Okay. So brainstem consists of the midbrain, pons, and medulla, or regulates a vital function such as breathing, serves as a sensory or motor filter for other parts of the brain. So midbrain receives and integrates auditory data, coordinates visual reflexes, sends sensory data to higher brain centers, right? This is essentially a control center to the upper, more complex brain centers. Pons controls breathing, which is here, this little bit. And then medulla oblongata, so controls breathing, circulating, swallowing, and digestion. This brainstem is considered the reptilian brain. Okay, so they sometimes call it the reptilian brain because reptiles lack a lot of the more developed brain centers that mammals have. Okay, whereas they have all of this. And it's interesting because there have been a couple of studies done where they've removed the higher level brain function. So they removed the cerebral cortex. Um, and I really don't agree with. Well, I don't know. This is an interesting study, but what they did was they actually removed um, the cerebrum and the cerebral cortex of a female cat, and it was a female cat. For some reason, males didn't have the same response, and maybe I didn't look too much into the paper to really identify the issue. But what they did was they completely removed the top part of the brain. They left the brain stem, and the female cat did all of the normal functions as long as it was kept in a cage. If you exposed it to other types of stimuli, it was unable to process that, and the cat would have some consequences with that. But if the cat had water, food, and was kept in a cage, it could eat, uh, 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 breed, it could defecate right normally, and it looked like a normal cat. And this is what the paper was kind of recording, saying that the brainstem is really all you need to survive for the most part. Um, Mind you, that was an immense amount of surgery that took place to remove this cerebral cortex of that cat. But yeah, not it was yeah, kind of an ugly study to read because it literally talked about the removal of a brain of a cat, which is kind of sad. And they just left the brainstem. But again, you really just need the brainstem to function in order to survive. That eating, breathing, mating can all be controlled here. These are all the brainstem activities. Okay. Cerebellum coordinates body movement. This is where you have a muscle memory, right? So if you practice something a million times, you try to get your golf swing down, or your tennis swing, or badminton, or whatever racket sport you play. Even if you don't have a racket sport, this is training for football or running or basketball. It doesn't matter. This cerebellum will allow you to have those fine motor movements and memory, right? This is muscle memory, ready to go. Okay. All right, so you have the right and left hemispheres. You also have the corpus callosum. The corpus callosum is incredibly important to control or to connect uh, the cerebral cortex together, right? Your two halves of your brain together. Um, this is where they always say that females are much better at males at multitasking, right? Because they have a thicker corpus callosum. So their connection between the two halves of the brain is thicker than males. And this is where they get that idea that women are better multitaskers than men. They just have a thicker connection. They can both sides of their brain can talk to each other at much quicker levels than males uh, because of the corpus callosum. Not saying that's true. I'm just saying that's what they say. So individuals attribute that to. Um, there have been cases where individuals do sever or cut the corpus callosum. This is to control for seizures. So in some very rare instances, people have developed incredibly horrible seizures that develop in one side of the brain. They'll transfer over to the left side and cause even worse seizing event. What they've noticed or what they've done, certain physicians will cut the corpus callosum in order to prevent the seizure from spreading to the other side. Okay. And this works in some cases. But you also have prevented or limited the both sides of the brains from talking to each other as well. So you have some interesting pathologies that are seen after the surgery. Many times you have the inability to perceive something with one eye versus the other eye. Okay. Right. You'll be able to see a color, but not be able to articulate it with your mouth. Right. You need both eyes in order to see that. Um, because one side of the brain will be color oriented, whereas the um, other side of the brain is going to be more verbally oriented. So there's a lot of interesting cases where people have had their corpus callosum severed, and they need to use both eyes in order to interpret something entirely. Right? If you close one eye and look at it, the object, you might only be able to see one facet of that. Whereas if you had the corpus callosum, you'd be able to see both facets with just one eye. Again, because your whole brain can talk to each other or can talk to itself. Whereas if it's severed, you lack the ability to share information rapidly with both sides. Okay. So here you have different areas of the brain. You have the frontal lobe, frontal association area, speech, motor cortex. somatosensory cortex, speech, taste, reading, visual association area, vision, hearing, auditory association, smell, temporal lobe. You have memory. Somewhere right here is the hippocampus, right? Um, so Fiend's Gage accident. So Fiend's Gage holds a 3-foot railroad spike that punctured his brain in 1848 left. On the right is one of numerous computer simulations of this accident depicting the frontal lobe damage, such an injury could account for Gage's personality changes after the accident. Okay, so railroad spike removed from Gage's brain. The railroad spike entered Gage's left cheek and exited through his skull. Okay, so you see here the traumatic injury of poor Gage here. And what is essentially occurring too when they mentioning the frontal lobe damage. A lot of individuals that get that receive injuries, traumatic brain injuries, or even where pieces of brain are removed or damaged. Many a times their personality does change. They can be more prone to spontaneous bits of anger or bouts of aggression. Um, they're not able to control certain aspects of themselves. They might be consuming more of a certain type of food. They may not like certain things anymore. There might be personality changes, right? And it's all due to the brain damage, right? The brain is changed either biochemically or even mechanically, like this. You do have changes in personalities. You have changes in normal, what can be considered normal for that individual. Okay. Um, and this is essentially why they have brain surgeries while the patient's alive or awake. Sorry. So sometimes the individual that's having the brain surgery will be playing an instrument that they know, or they'll be painting or writing something because it's something they would like to maintain. Because as that physician is going in and removing the brain tumor, they could be taking too much brain out, and then you're cutting or removing out those neuronal paths that you have and that you want to maintain, right? So brain surgery can be very tricky for this reason, as well as traumatic brain injuries or mechanical traumatic brain injuries can be difficult to kind of adjust or treat for because you never know, right? You could have damaged something that's so crucial to that person that makes that person who they are. It can be very difficult finding a new normal, finding a new baseline, right? Because the organ that kind of controls who you are is going to be destroyed. So, and this is how we get in. This is where we get into neuroplasticity. So the brain's ability to recognize neuronal connections throughout life, caused by normal development, changes in behavior and develop an environment and injury. Okay, and here's where we have the neuroplasticity with people that are able to survive severe brain injuries, or having their corpus callosum cut, or having one of their hemispheres of their brain be removed. And again, neuroplasticity kind of points out to the fact that individuals can live with half a brain and be nearly normal. Okay. Um, I don't know if the case was in the United States, but there was one individual who had half a brain. And the reason why they had half a brain was because they had severe seizures and early on in childhood. So the doctors made the drastic decision, and the parents, of course, to remove half a brain to save the child's life. And they did. And that child ended up developing, through after puberty, after various incidences in his life, with half a brain, and he was essentially normal. His brain, that half the side of that brain took over normal, functioning, and of course, one side of the brain controls different things than the other. So of course, there was limitations on certain things, but I believe that individual was either a math professor or math teacher, but I'm pretty sure if I'm not mistaken, that individual was a math professor at a university. So he was intelligent enough to teach mathematics at a university, which is insane, right? Because we you know some people have that joke where you have a half a brain. Well, half a brain is enough. So here we have hemispherectomy, which is going to be the removal of one half of the brain. And again, this is very, very, very rare. Um, I've only heard of it with individuals who have severe, severe seizures. So individuals have seizures that originate from one side of the brain that travel to the other, or just it just will not stop. With that, the neurons from firing on that one side or creating that seizure occurrence. And again, this will be a very drastic measure to remove half a brain. That's just scary, right? But again, this was in a child. It was early on. The doctors made the neurosurgeons made that call to remove half a brain. Um, and again, that child or that individual developed just fine. All right, so neurological diseases. We have Alzheimer's disease, a form of mental deterioration or dementia characterized by confusion and memory loss. So this is formed by you have tau protein, as well as I can't think of the other protein. Is it beta amyloid plaques? Yeah, beta amyloid plaques and tau protein. Okay, is it beta amyloid? That was something different. Definitely tau protein formation. So essentially what you have is you have accumulation of these proteins in the brain that prevent normal functioning. Okay. And it occurs in older individuals typically. You can have early onset Alzheimer's disease. Again, that's more genetic than anything. But again, it's the accumulation of these proteins in the brain that start forming uh, these plaques that stop normal neuronal functioning. Okay, so you have the memory loss and deterioration to dementia. Chronic traumatic encephalopathy, or CTE. This is a degenerative disease leading to the deterioration of a brain caused by brain injuries such as concussion, right? And this is terrible. CTE, there's been a lot of news recently on CTE, especially with a lot of the football players, some of them committing suicide, some of them performing um, really horrible acts of violence, and a lot of the times they're pointing to the CTE, traumatic brain injury, um, as a culprit of why their personalities have changed so much. um, and not to say that's the reason for everything or all the issues that they have, but this is not not an unknown thing, especially in the boxing community, right? Um, so Muhammad Ali, I mean, if you've seen Muhammad Ali after fighting and how severe his condition was, where he would shake, he lost a lot of his ability to communicate. You know, this is a case here with the CTE, and people just would say, oh, they're punch drunk, and not make a big deal of it. But specifically with boxers, everyone kind of knew, well, you get hit so much, something's going to be wrong with your brain. They never really thought about it with football too much until recently. And again, it could be a lot of the stigma associated with football. Well, you have a helmet, it's okay. Kids play this game, it's fine. And in reality, a lot of these kids, or a lot of these individuals playing football, are getting hit a lot harder and more often than some of the boxers were, right? Um, so yeah, it's a serious disease, serious illness, and again, you form these plaques, you form these occlusions in the brain, and the deterioration of brain occurs. And again, it's caused from the smashing of the brain inside the skull. It's almost similar to a concussion, right? You have little mini concussions all the time. Your brain's just sloshing around being destroyed. Okay. So major depression has been caused by CTE, they believe, right? What is it, Junior Seau? There was someone that the NFL football player, he actually killed himself. He was so depressed, um, and the way he killed himself was so controversial because he actually shot himself in the chest, right? And in doing so, he kind of pointed to the fact, or he left a note saying like he wanted his brain to be observed because again, he was different after football, different after all the concussions. Um, but major depression is depressive mental illness characterized by present sadness and loss of interest in pleasurable activities, right? And there's clinical depression. You can actually see this in a brain scan, right? Bipolar disorder, so depressive mental illness characterized by extreme mood swings, also called manic depressive disorder. Okay. Um, yeah. So brain activity. So in the purple And pink colors in these computer images indicate areas of low brain activity. Notice that the brain of the depressed person shows decreased activity in certain areas of the brain. So area of decreased activity of the brain, this is a depressed person. So here we have healthy person. So areas of normal activity, right? So area of decreased activity, and then boom, healthy person here. So you can actually see um, depression in CAT scan. All right, I think we'll stop there because we're getting into. Yeah, we'll stop there. Okay. Any questions? All right. So just a reminder, please register for your presentations. Okay, I'll send an email out in a minute. Um, I have extended the deadline for all the assignments on your McGraw-Hill and the assignments on Canvas till the 30th. Okay, so please, I know there's a lot of assignments, but please get these done. Okay, um, it's just gonna help your grade, right? It's not gonna hurt you to get these done by the 30th. Okay, so if you're struggling, if you feel like you're lacking some points, make them up now. Okay. Um, so I also opened up these exam questions. This will help you for the final as well. So complete these if you haven't done so already. So, right, we have 13 submissions here. Okay, I'm missing just about 10 individuals from the class. So just submit something, go through. And if you didn't notice, that assignment was actually all of test number 4, right? Or I'm sorry, all of test number 3. Those last questions. Also, make sure you are looking at the due dates for the term paper, and you can start submitting early, okay, to check for your plagiarism, right? Um, if you have any questions about this, I'll be going over this on Monday. Hopefully, someone or everyone starts registering for the dates of presentation, okay? But again, the presentation is going to be fairly simple. 10 minutes, it's going to be on your topic. I'm going to ask you 1 or 2 questions as well. Okay, and that'll give you a chunk of your grade there. So 10 to 20 points essentially, right? So look at the rubric. Look at the rubric, and it'll tell you exactly what you need to be putting into your presentation. So yeah, if you have any questions, please email me. I do have office hours today. It is for my bio 40 course, but you're more than welcome to drop in as long as you send me an email. I can send you that link. But for the presentation, okay, so I know presentations are starting Monday. Hopefully someone registers, but the oral presentation, again, it's going to be clarity, quality of content, organization, and then for the Q&A. So I'll be asking 2 questions, okay? So clarity, quality, and organization. So remember, you have a 10-minute spiel. You tell me about what your research paper is on, and then I'm going to ask you a couple questions, okay? All right. And that's it for today. Any questions from those of you who showed up? Only the 2 people, as usual. Nope. Okay. Well, I will see everyone on Monday, hopefully. Again, I apologize for the absence yesterday. That my power was completely out, so I had no internet. I was using my phone to send emails out, which is I can't use Zoom on there. It's just not enough. I don't have good enough data for that. But yeah, I will see everyone Monday.